Module 3, Hershey and Chase experiment from the chapter Molecular Basis of Inheritance. In 1953, Hershey and Chase confirmed DNA's prominent role in genetics by demonstrating that DNA is the genetic material of a virus called phage T2. The phage which infects E. coli consists of head, sheet, tail, and base plate made up of different proteins. DNA is packaged within the head of the virus. When phage T2 comes in contact of E. coli, the phage attaches to the bacteria by its tail. Then the phage injects the genetic material into the cell. The genetic material directs bacterial enzyme to produce viral offsprings. When the life cycle is complete, 100 to 200 progeny phages have been assembled inside each bacteria. The bacterium breaks open by lysis and the phages are released. Since the T2 phage consists of only DNA and protein, Hershey and Chase reasoned that the genetic material must be one of the two components. So, they designed an experiment to determine which it is. Further in experiment, the scientists prepared T2 phages which either have radioactive DNA or radioactive proteins. To make the component radioactive, Hershey and Chase infected E. coli with T2 and grew the bacterial cells into two different culture areas, one containing the radioactive isotope phosphorus 32 and other containing the radioactive isotope sulfur 35. All the phages released from the bacterium grown in the phosphorus 32 medium had radioactively labeled DNA, while all the phages released from the bacterium grown in sulfur 35 medium had radioactively labeled proteins. In the next phase of their experiment, Hershey and Chase infected E. coli with the two types of labeled T2. They then used a blender and a centrifuge to separate the proteins part of the phage called phage hosts from the rest of the infected cell. The scientists discovered that the labeled DNA appeared in the host cells but not in the phage hosts, while the labeled proteins appeared in the phage hosts but not in the host cells. In addition, the progeny of T2 labeled DNA were radioactive while the progeny of T2 labeled protein were non-radioactive. Now, because the genetic material is passed on from parents to the offsprings, Hershey and Chase concluded that DNA had to be the genetic material of phage T2. Their results had to convince the scientific community that DNA was the hereditary material. I hope you liked the video. Thank you.